narasumber spesial ya dari luar, yaitu pertama ada Pak Claudio Ciofi, Mr. Claudio Ciofi, come from Department of Biology, University of Florence, Italy. So I'm a big fan of Italian football, Mr. Ciofi. So it's a really honor. And the second speaker is Alessio Lanucci from the Department of Biology, University of Florence, Italy as well. So Mr. Ciofi will talk about from multi-locus genetic analysis to whole genome assays for wildlife population studies. And Mr. Lanucci will talk about the assessing population structure using multi-locus and genomic analysis, the Komodo dragon as a case study. Dan hari ini akan dipandu oleh moderator dan juga narasumber, yaitu Ibu Evi Ayu Arida dari Pusat Riset Zoologi Terapan dengan topik yang akan dibawakan yaitu pemanfaatan spesies biawak di Indonesia. Sebelum kita mulai, seperti biasa, akan ada opening remarks dari Kepala Pusat Riset Zoologi Terapan, Ibu Delicia Intarahman. Waktu dan tempat kami persilahkan. Mic-nya
Dari sini aja ya.
Aziza.
Oke, Assalamualaikum. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. First, welcome to Research Center for Applied Zoology Summer School, Series 4. This is our regular seminar, actually, that we have every 2 weeks. And today we really, this is our privilege to have with us two of our distinguished guests from Department of Biology, University of Florence, Italy, Claudio, and also Alessio. Thank you for coming and the opportunity to hear your expertise and sharing with us. Getting to us this session, we have our colleague, Bu Evi, that will lead the presentation today and also give us the perspective of pemanfaatan spesies biawak in Indonesia, offering us a unique perspective of on the utilization of monitoring species in Indonesia. We hope this sharing session will be an opportunity for fruitful discussion, networking, and also a chance to enhance our collective understanding of these critical topics that both of you will give to us. Thank you for being here with us today, and let's make this inspiring and enjoyable experience for everyone to enjoy the session. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Terima kasih Bu Kapus atas opening remarks-nya. Baik, selanjutnya kita akan lanjutkan kegiatan. Kita berikan kesempatan untuk moderator hari ini, yaitu Ibu Evi Ayu Arida, untuk bisa memandu acara hari ini sampai pukul 15.30. Jadi ya, sampai 15.30, 13.30, 14.30, 15.30, silahkan. Jika ada waktu nanti diskusi mungkin kita berikan waktu sekitar 30 atau 45 menit, sehingga nanti misalkan ada pertanyaan dari Zoom ataupun dari luar, nanti bisa ada kesempatan juga. Baik, tanpa membuang waktu, kami berikan kepada Ibu Evi Ayu Arida untuk memandu acara. Silakan, Bu.
Terima kasih, Mas Bram. Assalamualaikum warahmatullahi wabarakatuh. Yang terhormat Kapus Zoologi Terapan, teman-teman memandu acara Summer School namanya sekarang. Kita kedatangan 2 orang tamu. Ini ada Pak Profesor Claudio Ciofi. Ini dulu adalah pembimbing S3 saya. Memang saya melakukan S3 di Jerman, tetapi labnya di Italia. Betapa indahnya dulu ya, apalagi di Firenze. Kemudian ini Mas Alessio. Mas Alessio adalah periset pasca doktoral, mantan mahasiswa S3-nya Pak Claudio juga. Jadi kita satu alumni tapi ya beda hitam dan putih. Anyway, so pada kesempatan kali ini temanya adalah tentang biawak. Nah, jadi saya akan sedikit memperkenalkan apa itu biawak. Mungkin Bapak Ibu, teman-teman sekalian sudah pernah melihat biawak, tetapi saya akan merefresh tentang apa itu biawak. Pak Claudio nanti akan berbicara tentang pemanfaatan teknik molekuler untuk studi satwa liar. Kemudian Mas Alessio akan lebih mendalami lagi dengan contoh-contoh biawak yang hanya ada di Indonesia, yaitu biawak komodo. Baik, izinkan saya untuk share. Saya tidak akan lama, presentasi sekitar mungkin 8-9 menit. Kemudian dilanjutkan Pak Claudio sekitar setengah jam, Mas Alessio mungkin 20 menit saja. Lalu pertanyaan akan kita tampung di akhir 3 presentasi kita kali ini. Izinkan saya berbagi layar saya. Sudah terlihat belum ya Bapak Ibu ya?
Belum, Bu.
Oh, belum ya? Sekarang sudah terlihat?
Oke.
Baik. So, biawak. I will be speaking in Bahasa Indonesia, and in the meantime, probably I will change into English, but my slides are in English with pictures. So, biawak, ini adalah salah satu jenis satwa liar di Indonesia yang barangkali tidak banyak kita tahu. Kita lihat sehari-hari di belakang rumah atau di sekitar pemukiman, tetapi tidak banyak yang ingin belajar tentang biawak. Nah, saya tertarik. Mengapa? Karena ini adalah satu hal yang berlawanan, paradoks antara hama dan komoditas. Ini menarik bagi saya karena sebenarnya mengelola hama itu yang terbaik adalah menjadikannya sebagai komoditas. Jadi ekonomi sirkuler. Kira-kira gitu ya, Mas Ujang ya, jangan perak-perok ya. Nah, ini paradoks antara hama dan komoditas. Nah, bicara tentang biawak, ini adalah contoh-contoh ekstrim biawak yang mungkin tidak banyak kita kenal. Ya. Yang di sebelah kiri itu biawak komodo, saya foto di Loh Liang, kalau nggak salah. Tapi mungkin di Rinca. Yang di sebelah kanan itu juga disebut biawak. Ini mungkin untuk Bu Kapus ya. Ini biawak Kalimantan. Hanya ada di Kalimantan. Biawak komodo hanya ada di Nusa Tenggara Timur. Dua jenis ini di famili atau suku yang berbeda, tapi disebutnya oleh orang Indonesia sebagai biawak. Nah, ini kemudian gambar biawak air. Nah, inilah yang kita ketahui di Jawa. Jadi banyak orang Jawa yang hanya tahu ini saja biawak di Indonesia, padahal banyak sekali jenis biawak. Ini adalah paradoks. Hama di peternakan, unggas, hama di perumahan, pemukiman, tapi juga menjadi sumber industri kulit eksotis yang omsetnya bisa MM-an gitu ya. Mungkin teman-teman yang ex KLHK tahu ini betapa income PNBP dari KLHK tergantung juga dari jenis ini. Kemudian ini adalah contoh di perumahan saya di Kota Bogor. Bisa terjadi hal seperti ini. Jadi biawaknya ingin masuk ke rumah karena rumah ini mengandung mangsa biawak. Jadi di perumahan saya ini kondisinya seperti ini. Ada sungai kecil, kemudian ada bantaran yang rimbun, ada juga bantaran yang tidak ditanami atau tidak ada tumbuhan. Disitulah biasanya kita bisa melihat ada biawak yang nongkrong-nongkrong di situ ya. Jadi habitat biawak itu semacam ini. Ini perlu kita ketahui bahwa yang menghuni perumahan tersebut adalah biawaknya dulu, bukan kita dulu ya. Apalagi bukan developer dulu. Ini kejadian ini tidak hanya di Indonesia, ada juga di Malaysia, di Thailand, bahkan di Singapura yang negaranya sangat super bersih dan higienis. Ini contohnya ada biawak air yang melintas di sirkuit F1 sehingga balapan sirkuit F1 harus berhenti karena ada yang di Pertuan Agung biawak yang lewat. Ya, kira-kira begitu. Yang akan saya ceritakan kali ini hanya satu saja, satu bagian. Ini slide saya sebenarnya saya pinjam dari waktu melakukan implementasi teknologi. Jadi saya mendaur ulang slide-nya. Mungkin saya hanya akan memberikan sedikit gambaran mengenai pemanfaatan biawak di Indonesia. Yang kedua ini, disinilah sebenarnya letak paradoksnya. Jadi berburu itu mengeradikasi, membuang, tapi bagi kami periset, kita periset sebenarnya adalah memperoleh sampel, gitu ya. Ini namanya Waran atau dalam bahasa Jerman atau Wakol dari bahasa Arab artinya monitor. Lalu mengapa dalam bahasa Inggris biawak disebut monitor lizard karena dia suka memonitor, gitu ya. Bukan monitor komputer ya. Ini pertama kali dicetuskan istilahnya atau nama genusnya oleh Merem di bukunya yang berjudul Versuchene Systems Amphibien. Jadi reptil seperti Varanus ini waktu itu masih disebut sebagai amfibi. Jadi amfibi sekarang hanya katak saja, tidak ada kadal. Kemudian studi yang lebih terkini ya tahun 2012 ini sebenarnya sudah cukup lama. Ini memetakan hubungan kekerabatan antara semua jenis Varanus di dunia. Jadi di sini bisa terlihat bahwa Varanus ada di Australia paling banyak, di Indonesia nomor 2, di Afrika paling sedikit. Biawak di Eropa misalnya sudah punah dan di belahan dunia yang lain bahkan tidak pernah ada, seperti di Amerika Selatan. Ini mungkin Bapak Ibu akan sedikit terkejut kalau belum pernah melihat ini bahwa industri kulit eksotis ini banyak di negara-negara Eropa. Contohnya di Italia ya, rumah-rumah mode Italia itu mengambil bahan baku kulit biawak dari Indonesia. Selain juga kulit ular. Ini contoh-contoh yang murah saja, Pak Ibu. Jadi dompet semacam ini, kalau ini mah harganya cuma 70.000 ya. Tapi di sini harganya bisa sampai 1 juta lebih. Ini contoh di marketplace. Kemudian ini contoh lagi bahwa biawak ini menjadi ikon fashion yang mewah ya. Ini dilelang di Balai Lelang Christie tahun 2021. Akhirnya terjual dengan harga 62.500 dolar Amerika. Jadi ini dari 1 ekor biawak dengan harga begini ya. Padahal kalau kita sampling mungkin beli dari pengepul harganya mungkin hanya 50 sampai 100.000 aja. Kemudian selain sebagai komoditas kulit, biawak juga sudah dimanfaatkan sebagai komoditas pet atau hewan kesayangan. Jadi seperti saya katakan di awal tadi bahwa pemanfaatan biawak ini selain untuk bahan baku kulit eksotis juga menjadi sumber hewan kesayangan eksotis. Ya. Ini contoh lagi di websitenya Exotic Pet Decor. Ini bermacam-macam jenis biawak air. Sebenarnya bukan bermacam-macam jenis, tapi bermacam-macam morph ya, bentuknya macam-macam. Ini ada yang modelnya sulfur, ada yang modelnya dan lain-lain seperti ini. Kalau ini contoh yang lain, biawak selain biawak air, ini hanya ada di Pulau Biak, biawaknya. Ya, Biak di Papua, sebelah utara. Ini biawak hijau, sangat cantik, dan ini juga diperdagangkan sebagai komoditas hewan kesayangan ekspor. Ini saya temukan di Instagram, ini ada seorang kolektor di Amerika Serikat yang membuat rumahnya menjadi kandang biawak seperti ini, sangat mewah. Jadi ini kandang yang di sebelah kanan ini untuk didiami oleh pet atau hewan kesayangannya berupa biawak hijau. Asalnya bisa dari Papua biasanya. Jadi sebagai ringkasan, ada 30 jenis biawak di Indonesia dengan distribusi yang tidak merata. Jadi sebarannya di Indonesia, saya nggak punya ini ya. Di Indonesia bagian barat itu yang di sebelah kiri garis kuning atau garis Wallace itu keanekaragamannya sangat sedikit. Kemudian yang di sebelah kanan garis kuning jadi di Indonesia Timur jenis-jenisnya lebih beragam dan banyak endemisitasnya di pulau-pulau kecil pada khususnya. Ada 30 jenis, tetapi kebanyakan jenis dideskripsikan atau ditemukan di Indonesia bagian timur dengan endemisme yang cukup tinggi juga. Oh ya, jangan lupa juga selain 30 jenis varanus, ada satu jenis biawak unik, biawak Kalimantan, ini di suku yang berbeda, Lanthanatus bornensis. Ini juga menjadi salah satu komoditas pet, bahkan diselundupkan dengan cara yang sangat lucu biasanya ya. Ada yang disembunyikan di celana, ada yang disembunyikan di koper, dan sebagainya. Jadi sebagai apa? Sebagai final words mungkin ya. Ada 3 pemanfaatan biawak di Indonesia. Satu sebagai sumber kulit, bahan untuk fashion, sebagai hewan kesayangan, dan juga tentu saja ini komodo sebagai ikon pariwisata premium di Indonesia. Jadi semua biawak tidak ada yang tidak berguna. Sebenarnya disebut hama itu karena sudut pandangnya saja. Demikian yang bisa saya sampaikan perkenalan tentang biawak jenis-jenis biawak Indonesia ya, ada 30 plus 1. Selanjutnya saya akan stop share karena Pak Claudio akan mempresentasikan apa topiknya, yaitu I'll switch to English, from multilocus genetic analysis to whole genome assays for wildlife population studies. So I'll give you about 30 minutes, Claudio.
Ya, terima kasih. Ibu. Tapi tanpa kesan, program, dan terima kasih telah mengundang kami pada presentasi saya di bahasa Inggris. Maaf. It will screen there. Oh, we're doing from here. Oh, we're doing from here, but I don't know what's into full screen. It's full screen.
Enggak, full screen.
Okay.
Yeah, perfect.
Okay.
And where is the screen?
Share screen.
It's already shared.
I can't see. Oh, okay.
Yeah.
Okay. So I will, in this presentation, I will talk briefly about the original methods that are used for studying population structure, original methods based on DNA analysis. You can see an example on the left, lower left part of the slide. And then I will introduce some more advanced techniques dealing with whole genome sequencing, of which you can see an example to the lower right of the slide. So when we study population, wildlife, natural population using molecular markers, we generally talk about, and we're talking about animals, okay? We talk about, or plants, we talk about organelle DNA, so mitochondrial DNA for animals, and chloroplast DNA for instance, plants, and nuclear DNA. The classical example that we know when we use single or multi-locus molecular markers are, for instance, the characterization of a section, a part of the mitochondrial genome that can be compared among different samples, among different individuals, for instance, of a population. Here we have an example of 3 individuals that have which the first 2 individuals have the same sequence, the same mitochondrial sequence, and the third individual have a substitution, a 2 substitution where the arrows are pointing at this sequence. So the classical technique to use to characterize part of the mitochondrial genome, for instance, when we talk about intraspecific level, so comparison among populations which are very close to each other from a phylogenetic point of view, one classical marker is the control region, which is the region that does not codify for genes it's the only region of the mitochondrial DNA which does not codify for genes. So in this case, we use a PCR to amplify this region, and then after amplification of the control region, we use generally the cycle sequencing or chain termination reaction, and after the chain termination reaction, we run the products via capillary electrophoresis, and the results basically are the electropherograms like this one, in which one can see the sequence, in this case, of 4 individuals, in which the first individual has 1 substitution where the line, the central line is pointing, with respect to the other 3 individuals that have the same, exactly the same sequences. Okay. So this is the classical method from the electropherogram that can also, they had to be also manually curated, because sometimes the software that is linked to the automated DNA analyzer does not recognize some peaks, or there are peaks that overlap with other peaks that represent nucleotides. So one had to go in this position and manually edit the sequence to make it right. So from these electropherograms, then basically these electropherograms are translated into text sequences, and these sequences are compared to each other to provide a number of information. Here I give an example, classical example of how one can use these markers of the mitochondrial DNA to assess difference among island population this is our case study of Komodo dragon. Here a classical table in which to the left you can see the number of haplotypes, so the number of unique sequences that have been characterized in the population, the numbers of the location refer to the is arbitrary, is the number of correspond to the locations where these haplotypes are found, and is the number of individuals that have been sampled, and the segregating site or the polymorphic site are the site along the contour region from site 1 to site 700 is where these haplotypes differ between each other. Okay, so on the basis of this information, one can basically reconstruct what can reconstruct the phylogeny of a species. And in this case, we are talking about intraspecific phylogeny, so difference among population. And you can see here, for instance, that see if I can get the laser point. The different colors correspond to different haplotypes, and the size of each part of these the slice basically correspond to the number of animals that carry a specific haplotype. And using this marker, one can, for instance, determine the difference from the island of Komodo in Komodo National Park, the group of Rincha, West Flores, Guillemotang, and Musacode. And if you go and if you look here on the bottom, the part of North Flores that have different haplotypes from the rest of the archipelago. One thing to notice here is that with this marker, it is difficult to recognize and to assess the difference between close population, like the island of Rincha and the island of West Flores. So based on these, on single locus marker, the mitochondrial DNA in this case, we won't know whether an individual comes, for instance, from Rincha or comes from West Flores. An example of nuclear DNA markers that are, on the other hand, markers that are based not on the difference in sequences, but they are based on the difference in allele lengths. This is two samples which have two alleles, of course, because these markers are inherited in a Mendelian fashion. So one from the mother and one from the father. And the first samples is heterozygous for allele 1 and allele 2, and the second sample is homozygous for allele 1 and allele 2. Here we have three different alleles, okay? And these alleles are characterized basically by difference in the number of co-repeats of the microsatellites. In the sample number 1, we have the first allele have 11 repeats, the second allele have 9 repeats, and in sample number 2, both alleles have the same number of repeats. This is the classical section of microsatellites. And this is how one characterizes the microsatellite locus by PCR. This is species A, and I put species A because in a while I will explain a relatively important thing about using species-specific PCR primers. Okay, this is very important. In this case, we have allele 1 and allele 2 on one animal. Of course, these are double because we are talking about double-stranded DNA. Forward primer, reverse primer, forward primer, reverse primer that basically attach to alleles, and by PCR, they amplify allele 1 and allele 2. The forward primers is marked with a fluorochrome of, in this case, FAM, which has a blue color. Okay, we generally can use a maximum of 5 different colors, 5 different fluorochrome to mark the forward primers. And the results of these PCRs is basically after these PCR products are run through a electrophoretic system. Here you see the capillaries of these. This is an ABI 3130, which is a relatively old machine. I saw in the genomic laboratory you have a more recent capillary electrophoresis system. The results of the amplification of those two alleles that you've seen before are these two peaks. One is 98 base and one is 100 base, and these basically are the difference between these is basically 1, 2, 3, 4. There are 5 repeats of difference, 5 dinucleotide repeats of difference between these two alleles. The important thing of using species-specific primers is to avoid incidence of null alleles. So if I use, for instance, the primers that have been developed for species A, and I use them on species B, for instance, another species of varanid, say the water monitor, okay? If I have on the second species, on species B, if I have a mutation at the points where the primers find complementarity, and I have a mutation both in allele 1 and allele 2, basically the PCR doesn't work because the primers do not attach. So I have a null result, okay? Which is okay. It means that these primers that I developed on species A do not work on species B. The problem comes when these mutations are not on both alleles, okay, but are on only one allele. So in this case, only one allele is not amplified, the first one, and the second allele is, on the contrary, amplified because it doesn't have mutations. Here I missed a cross, it should be a cross here as well, okay? So in this case, this individual that for these two alleles, this locus is heterozygous in reality, my result is basically this, only one peak. This peak basically doesn't appear. So it's a fault homozygote. And this is quite important because if I miss an allele, I can basically misassign an individual in one population or the other. And this is very important also in parentage analysis if one allele is missing. So it's very important when working with macrosatellites to use species-specific primers, PCR primers. The allele scoring is basically done with automated software, for instance, like GeneMapper. This is an example of 3 loci. We see 3 different colors. So the rows are different individuals and the column are 3 different loci in which the forward primer has been marked with a different fluorophore. Okay. The result of this is a multilocus genotype. So for each individual of different locations here in the row, I didn't put any more 1, 2, 3, 4, 5, but it's self-explanatory, I think. We have a multilocus genotypes. This is for 16 loci, okay? And for 16 loci, we have allele 1, allele 2, allele 1, allele 2, and so on. And from these, table, one can perform similar analysis of the one that I showed for the mitochondrial DNA. This is an analysis based on 18 microsatellite loci. This is a collaboration with the late Ibusri and Pazen several years ago, in which you can see that we have basically the same situation that we saw with the control region in the mitochondrial DNA, apart from the fact that here is shown in red, okay, the value of the fixation index, for instance, FST, which is an index of population diversity between Flores and northern Rincha, is, so between this part here and this part here, that with the mitochondrial DNA could not be told apart. In this case, this value is slightly significant, statistically significant, with 1 asterisk here, okay? So that means that with microsatellite, we go a bit farther than the single genome mitochondrial DNA, but we are not there yet, because this difference is not very, very striking, very strong, okay? So we need something more. than a single locus or a multi-locus mark. This is another type of analysis based on Bayesian statistics, in which there are a number of possible clusters of genotypes that are for which the likelihood is assessed, and in this case, 4 cluster and 5 cluster possible cluster have the same likelihood of existing. And the cluster here are indicated by different colors. So on the top here we have 4 colors, say 4 clusters, that identify the difference between the island of Gili-Motang, the island of Komodo, the island of the part of North Flores, which here is not shown in the map, and then a cluster that comprise the little island of Nusa Code, the island of Rincha, and West Flores. With k equal to 5, we can start seeing a slightly different of West Flores, but still in Rincha there are some mixture of individuals that belong both to the 5th cluster and to the 4th cluster. So again, with this analysis, we go a bit farther than mitochondrial DNA, but still we cannot reach the level of how can I say, the level of refinedness and detail that we need to distinguish, say, 2 very close populations, Rincha in this case and Flores. So point is to switch to whole genome analysis, whole mitochondrial genome, or whole nuclear genome, in which one can basically identify tens of thousands of single nucleotide polymorphisms that provide a better resolution. What I would like to, so basically, how does it work? Basically, we have to produce I'm talking about the nuclear DNA, not the mitochondrial DNA, because mitochondrial DNA is relatively easier than the nuclear DNA. We have to produce a number of sequences, okay, single sequences. They are called reads, okay. They are then assembled together to provide the entire sequence of the genome, okay. And this can be done in different ways. Can be done by using techniques that provide short reads, or techniques that provide long reads. And basically, to assemble, to obtain a genome is for the first time, if we don't have the genome of a species, for the first time is always best to use long read sequencing. You can see here this example with a puzzle. No, to reconstruct the puzzle is better to have large pieces rather than very small pieces. Okay, and there are generally 2 techniques. One is the PacBio techniques, and the other one is the nanopore. I saw in the genomic building you have here at Brin the nanopore. So I suppose that some of you have already used that, and so I'll just talk briefly about the Pacific Bioscience technology, which is also called single molecule real-time sequencing. This is a chip to the left. This chip basically has, if one goes inside this chip, you will find that these nano wells, okay, that are called zero mode waveguides, and the diameter of these wells in which the DNA molecule is inserted is very, very limited. So the diameter is so small that the light cannot pass through these well. So basically the diameter of the well is smaller than the wavelength of the light. So basically that means that the light remains, it only can illuminate the base of this well and not all the rest. So that means the if we put the nucleotide here, each nucleotide has a different fluorochrome or different color. All the nucleotides that are in the well, these are not detected. okay, because the light doesn't reach this level here. They are detected only when they are cooked by the DNA polymerase, which is stuck to the base. And when the polymerase take one nucleotide here, then the fluorochrome is detected by the light. Okay, this is the general, say, concept with which these machines work. And this, of course, is done by in millions of these nanowells, these zero-mode waveguides that are found on the chip that I've shown you before. Okay, and generally these are the kind of machine that are used. They're the same size of the Novasec 6000 that you have in the genomic build. Okay. So while for sequencing this genome you need the machine, the library preparation can be done in a standard laboratory. Okay? So the library preparation involve the production of these fragment of the DNA. So the DNA has to be fragmented before, but these fragments are not short fragment of 100 or 200 base pair, are long fragment of 20,000 base pairs. Okay? And the accuracy here is done because these fragments here, these fragments are attached to linkers here in blue, to the left and to the right, so they circularize the molecule. The molecule becomes a circular molecule. And then the polymerase, okay, it will sequence this molecule not one time, but several times. So we will pass more time on the same molecule. So if the polymerase makes an error the first time, the second time it won't make the same error in the same point. And the third time it will make an error on another point. So basically one will have several copies out of 15 copies of this molecule. They are called subreads. They are basically put together on a single molecule consensus sequence. So this single molecule consensus sequence will have a high accuracy, even if the length of the fragment is 20,000 base pairs. This is something basically that one cannot achieve with a nanopore, but can be achieved with a PacBio. Okay? So basically the library process here is this one. One has to, of course, assess the quality of the DNA. Okay, the DNA between 300 nanograms and 5 micrograms, so it's quite a lot. Okay, it's not a method that you can use with, say, non-invasive sample or with environmental DNA, but you need some relatively good sample, tissues or blood. Okay. And then the DNA has to be shared, so it should be fragmented into these pieces of around 20 base pair. When the DNA has been fragmented into these pieces that I showed you before, then the adapters are attached to these fragments, okay? And after these, we have a session of fragment selection through an automated DNA fragment pulse field electrophoresis system that you have. I saw it in the genomic laboratorium. This is a blue pipping, okay? So in this case, basically, in this way, one can set the size of the fragments that you need to be sequenced. And you can see here in these electrophoresis that after this selection, the fragments start from, say, a minimum of 10,000 base pair to a maximum of 88,000 base pair, and in this case from 5,000 base pair to a maximum of 31,000. This is an average size of 24,000. This is an average size of 10,000. So basically, the average size is between 18,000 and 20,000 base pair of these fragments. Okay. So once these fragments are obtained, then these can be frozen or kept at 4 degrees for up to 1 week. And then the polymerase is bound and the primers for sequences are bound. And then these molecules are inserted in those nanowells that I showed you before in the machine. So basically, if one doesn't have the machine, can construct this library up to this point, or up to this point as well in the laboratory, and then send the library for sequencing to a provider that has this pack value. Okay. So basically, this is, for instance, an example of the number of reads, of the distribution of the reads of a certain size. You can see this is an example for a human that goes from 10,000 to up to 40,000, and this is the distribution. So most of them are in the range of between 18 and 20,000. This is for the mosquito, and more or less has the same, a very similar results, between 10,000 and 30,000, a bit less, okay, but still the average is about 18,000. Okay, so this method works both for samples with lots of DNA, or even for smaller samples of smaller individuals like an insect that can be sequenced. So bottom line is that when all these reads are obtained, these reads are put together to assemble in an assembly for the genome. This genome that is produced by these long reads is generally used as a reference. Okay? This is called a reference genome. It's a high quality, nearly error-free DNA sequencing representing the entire genomic makeup of the species. There are several international projects that aims at producing reference genome for all biodiversity on Earth. The Earth Biogenome Project is one example. In Europe, we have the European Reference Genome Atlas and this Biodiversity Genomics Europe project funded by the European Union, again, to produce reference genome for all the species. So this is important because once you have the reference genome, then one can use resequencing, so can use a cheaper technique that is generally based on the production of short reads. And here comes the Illumina, which is the machine that you have in the genomic building. But this machine produces short reads. Short reads are not good to make a de novo assembly of a genome, but they're good if one have a reference genome that these reads can be aligned to. So once the reference genome is available, then one can use the sequencing by synthesis method to produce short reads and to align the short reads to the reference genome. And here quickly, if I have another 5 minutes of time, these again involve the preparation of a library. So fragment the DNA of your example. One have 100 individuals of a species of a varanid lizard that Ibu Evi has shown you before. Okay, wants to know the difference between population A and population B. So I have, for instance, 100 individuals from population A and 100 from population B. I have the reference genome of that species of varanids, okay? And I took my 100 and 100, so 200 samples. I extract the DNA and I fragment the DNA for each sample. Then I have to attach my primers that will be used basically for sequencing, and also some oligonucleotide. And these oligonucleotide that have the same sequence of oligonucleotide that are fixed on a flow cell, okay, where the sequencing will happen. Basically, once these fragments are ready with these with the oligonucleotide for the sequencing and the short oligonucleotides for anchoring to the flow cell, so basically it is something like this. Then these fragments, millions of these fragments, of course, they are inserted into the sequencer, and actually in the flow cells. The flow cells look like something like this. Okay, so we have, this is the flow cell, this is an enlargement of the flow cell, and if you zoom inside here, you'll see basically all these oligonucleotides sticking out of the flow cells. And these are the point of anchoring of the sequences that we've seen in the previous slide. So this sequence here, for instance, this sequence here, the one in red or in yellow, okay, will stick to one of these sequences that you have here in the flow cell. So you'll have lots of fragments that basically stick out vertically, and after these fragments are amplified, and after they are amplified, they form clusters that have the same sequence for each cluster, and each cluster then the sequencing begins. The sequencing by synthesis, in which the nucleotide are basically attached to these vertical fragments, and each time a nucleotide is attached, the machine register the fluorochrome of the nucleotide, and so record basically the sequence that the fragments that have been attached on the flow cell have. Basically, and these again happen in one point where one cluster of the same sequence has been formed, and then millions of points in which you will see all this color, and each color corresponds to a certain base that has been attached at a certain time. And the result are all these millions of sequences, okay? And these millions of sequences, of course, they have barcodes, and these barcodes belong to the different samples. So we will have 200 barcodes to distinguish those 100 samples from population A and those 100 samples from population B. So at the end of this process, basically you have all the reads for sample number 1, okay, that will be aligned to the reference genome. All reads from sample number 2, they will have the same. Reads for sample number 3, reads for sample number 4, and so on. And then when these reads will be aligned, basically for instance, 20 samples, we will start looking at the polymorphisms, no? At the SNPs, at the SNP calling that then Alessio will show you an example of this. It will help basically assessing the difference among samples, but not in one locus, not at 15 loci, but at tens of thousands of loci. So it will provide a much higher definition than the single or multi-locus analysis. So here I just put as a kind of a follow-up of these seminars in accordance also with Breen and when I had a chat with Ivo Evi, maybe for next year or wherever, when it's more convenient for you, is to basically provide a 1 week or 2 weeks kind of training sessions on some techniques that are of most interest here. For instance, I just put, for instance, the microsatellite analysis. If there is still an interest for these markers, microsatellite amplification, allele scoring after capillary electrophoresis using GenMapper, and population structure analysis, and or the whole genome sequencing library preparation. And these can be either the PacBio for reference genome, or better the Illumina sequencing by synthesis for population structure analysis. So DNA quality check after extraction. There are a number of kits, but the most common kit is the PCR-free library preparation for sequencing, so it doesn't imply using PCR. Sample pooling strategies, if you have, of course, more samples, and the multiplexing after sequencing, that means to then tell apart which are the sequences not from one sample, from the reads from one sample from the reads on the other. I put here also a WGS analysis that probably Alessio will talk a bit more in details. Sorry about if I went a bit long. Okay, thank you.
Thank you, Claudio. That was really heavy in this afternoon, tropical time. There's a lot to digest, but we have to listen to more details, unfortunately. And it's going to be a chance to learn something. Alessio is going to present his talk on assessment of population of Komodo dragon using genome sequencing.
Okay, thank you very much. I'm sorry I don't speak the language, so you have to hear me everything in English yet. So I will show you the results of our genomic approach to the study of population structure and demography in our case study, the Komodo dragon, which is also coming from collaboration with Ibuebi and Padzain from the Brin. Just a quick word on what I generally do in the molecular ecology lab of Professor Cioffi. We study population and conservation genomics of many endangered species. Some of them are Italian endemism, like as you can see here, some fish, mammals, and also Italian reptiles, but we also study tropical species as the Komodo dragon. As Professor Cioffi said, dealing with population and conservation genomics means also to deal with production of reference genome. So that is something we do in-house. And production of reference genome means also to have information on chromosomes. So that's why we also have expertise in cytogenomics. And lately, we are also applying molecular tools to the study of ecosystems using environmental DNA, which is a very powerful tool, actually. So we started our genomic experience with the Komodo dragon in 2019, when we first published the reference genome of the species, which is the starting point of the genomic analysis that I will show you. So this is the paper that we managed to publish in Nature, Ecology and Evolution, and it was one of the first genome that got published for squamate reptiles. We did that by using multiple technologies, multiple sequencing technologies, mainly based on long reads approach. As you can see here, we used both PacBio and Oxford Nanopore, and we use also BioNano and 10X, which are two other supporting technique at that time that we're using for the scaffolding approach. You can see here some of the statistics of the genome. You see that the assembly size was 1.5 gigabases. We had a pretty good N50 scaffold length, meaning that the genome was very accurate. And you can see here the reptiles genome that were published at that time, and they were quite a few. And many of them, as you can see, were not from squamate reptiles, but they were mainly, they were mainly from Archaeosauromorpha, meaning the turtles and the alligators and crocodiles. Now we have more genome of squamates also, luckily. So we had to produce the genome for our genomic analysis, but we also investigated some characteristic of the Komodo dragon from a genomic perspective, and we found something very interesting, which is related to the physiology of the species. For instance, we found an expansion of vomeronasal receptor genes, as you can see here, or maybe not. As you can see here, Komodo dragon has its own specific family of vomeronasal receptor genes. Vomeronasal organ is the olfactory organ that is mainly connected to the prey detection. So we connected this finding to the fact that Komodo dragon has a particularly sensitive olfactory system that is used for prey detection. And we also found a positive selection of coagulation cascade genes. And we link this to the fact that Komodo dragon's venom is an anticoagulant, which is also used in male versus male fighting. So basically, we interpreted this positive selection of coagulation cascade genes in a way that is basically connected to the resistance of Komodo dragon to its own venom, which was something very interesting. So anyway, genome was done. We had this very nice piece of information for the species, and we applied it 2 years later to the first population genomic survey of the species. This paper also got published in 2021 in Molecular Ecology. In a special issue dedicated to whole genome sequencing in molecular ecology, actually. And again, here was also one of the first study that used this approach in squamate reptiles. So just a quick overview again of the specimen that the samples that we used. We got, in this case, only 4 samples for each population. And we got 4 samples from Komodo, 4 from Rincha, 4 from Lusacode, 4 from Guillemotan, 4 from West Flores, and 4 from North Flores. So 24 samples in total. We sequenced this sample in-house. Likely we have the possibility to follow all the workflow in our lab, as we have genomic facilities there. So we sequenced our 24 genomes and we mapped them to our reference genome that now we had. Basically, as Professor Cioffi showed you already, this was the approach. So we mapped each individual to the genome, discovered the small differences that one individual has against the reference genome or in comparison to all the other individuals, okay? So we got this nice overview of all our data based on, look at here, 600,000 SNPs, which is quite different from the 17 loci of microsatellite, okay? Just a quick overview of the coverage that we have for each individual. The coverage is basically the depth of sequencing of each individual, meaning that when you sequence an individual, you do not sequence the genome just once, but you repeat the sequences a number of times. That it's the meaning of the coverage. So for instance, this individual has a 30x coverage, meaning that the individual genome was sequenced 30 times, okay? As you can see here, for each population, we had an individual with a higher coverage, and this, as you will see later, was done mainly because demographic analysis require high coverage. But the coverage showed us also some very interesting involved in sex determination. As you know, Komodo dragon has a sex determination system that is ZZ for males and ZW for females. And if you check the coverage of Z chromosome, which is what you can see here in this graph, you will see that the Z chromosome in males is coverage double the amount in comparison to females. And this is actually because of the sex chromosome system, because males had ZZ, so they have double the amount of Z chromosome in comparison of females, and that is reflected by the coverage. So this was quite important information to have for our dataset that come out easily from whole genome sequencing data. But let's have a look at the structure we found using our 600,000 SNPs. So as you can see here from this phylogenetic tree, which was done with BEAST, you can see that, again, North Flores is considered the outlier of our population. Then we have Komodo, and then we have a core group here made up of Guillemotang, West Flores, Rincha, and Nusa Kode. PCA told us the same. We have Komodo, North Flores, and the core group here, as well as the admixture analysis that identify, but here in the admixture analysis, you can see that already whole genome sequencing is starting to distinguish the population better, because you can see here, of course, North Flores and Komodo, but West Flores, Nusa Kode, and Guillemotang starts already to differentiate using this data. So it's confirmed what we found with the microsatellites, but I like to underline here that these results were obtained only with 24 individuals and not more than 100 as was done in microsatellites. So this is something that should be considered as one of the power of whole genome sequencing analysis, that you can have a very nice structure overview of your population using also just few individuals. But the structure, the population structure, is not the only thing that you can do with whole genome sequencing. Whole genome sequencing provide you data that are spread along the entire genome of a species. So you can do much more with that. And that is what we did, actually. Using whole genome sequencing data, we managed to have a very deep understanding of the genomic diversity of our population. As you can see here, these are our 6 population of Komodo dragon. This is the genomic diversity calculated based on Watterson theta estimates. Anyway, this is the genomic diversity. And as you can see here, Komodo dragon diversity is comparable or even lower than some other reptiles which are critically endangered or vulnerable. This doesn't mean that Komodo dragon is in bad condition. This is need the integration of also ecological data. But anyway, the results here says that the genomic diversity is something that should be, let's say, monitored during the in the wild populations. Here on the right, you can see the same things based split on neutral and exons region, and it's also informative from a conservation point of view. The last thing I'm showing you from this paper is this powerful analysis that whole genome sequencing allow you to do, which is actually, so now with the population structure and the genomic diversity, I have shown you what is currently going on in Komodo dragon's population. Whole genome sequencing data can also show you how was the demographic of the species in a very broad range of time. How do you see this plot here? Here you have, on the y-axis, you see the effective population size, and on the x, you can see years ago. So here you have today, and here you have 1 million years ago, which is half of the Pleistocene. It's quite a long time ago. And as you can see here, we managed to reconstruct the effective population size of all the 6 populations during this broad range of time. And as you can see here, by the way, as many other reptiles that were influenced from the global lower temperature that we had from the Pleistocene till now, the populations declined. okay? But apart from the results of Komodo dragon itself, you can see here how powerful a whole genome sequencing analysis can be, even in the description of the demography of the species. So, okay, fine. We got our first preliminary genomic assessment of population of Komodo dragon population based on genomic analysis. So what we did was to, but as I said, we did this only considering 24 individuals. So we said, let's see what happens if we consider 145 individuals using this approach. Now, to process 145 individuals with high coverage is quite complicated, even from a computational point of view. So what we did was to use a low coverage genome sequencing approach. So we sequenced the 145 individuals with a low coverage, and we mapped them to the reference genome. Just to be clear, low coverage is the approach that it is used lately to overcome the problems due to the computational problems due to have large amount of data for many individuals. So this is a very quick history of genomic approaches. In the beginning, we had RAD sequencing, which considered chunks of genomics to be analyzed in parallel. Then we had whole genome sequencing, or pool-seq, on which we based our first genomic study. And as you can see here, the depth of coverage was quite high. Now the main approach used in population genomics is the low coverage whole genome sequencing, where each individual is covered not as much as I showed you before, but still from here we can retrieve good information. If you want to have a look at this in details, I'm citing here this paper, which is very nice, A Beginner's Guide to Low Coverage Whole Genome Sequencing for Population Genomics, which is very informative. So what we found with this low coverage analysis, I'll show you here the slide that Professor Cioffi already showed you about the structure that we found using microsatellites. Again, you can see here that the micro were able to distinguish Guillemotang, Komodo, North Flores, but for the core group of Rincha, West Flores, and Nusa Code, the situation was a bit more confused. Instead, this is what we found with just a first scanning with a PCA of whole genome sequencing data. You can see here that the 4 groups, this is the PCA only with the core group, excluding North Florence and Komodo. We can start to see the differentiation of Flores West, Nusa Code, Guillemotang, and Rincha. So what we decide, so these analyses are still ongoing. We still need to check for genomic diversity. We still need to check for demography, as I showed you before. And this is something that is going on right now. But still, we decided to give it a try to this data already to produce a SNP panel, a panel of single nucleotide polymorphism that might be applied for the assignment of Komodo dragons, or anyway for the recognition of samples based on the population they belong to. So this is what we did. We selected out of more than 1 million SNPs, we selected only 96 SNPs, 20 SNPs basically for each node that separates the populations. And we perform again our analysis with these 96 SNPs. And this is what we found. We managed to distinguish clearly all the 6 populations. You can see that we still have some sign of introgression between Rincha and West Flores, but it is actually biological. So we probably must admit that, let's say, we probably need to deal with the fact that Rincha and West Flores used to have gene flow, so signs of admixture are still very evident in this population, but still you can see that they are quite very much distinguishable one another. So this is, of course, very relevant from a conservation point of view to be able to distinguish the 6 population one another. And from about this, I may leave the word to Professor Cioffi that can be more precise than me.
Yes, just briefly, the importance of distinguishing in this case Rincha from West Flores is because Rincha, you can see the dotted line around Komodo. Rincha is the boundary of Komodo National Park. Distinguish between a population that is part of the Kawasan, so Komodo National Park, and a population that is out.
Or you can use mine.
Yeah, so distinguishing basically the population of Rincha that is inside Komodo National Park and Flores, which is outside of Komodo National Park, is important to identify the origin, for instance, of a captive individual, which is a captive individual that belongs to the national park or belongs outside of the national park. And in case there are programs of demographic reinforcement of some small population in Komodo National Park, the fact that one can distinguish the two population, protected area and non-protected area, can actually determine whether it is really worth to take animals from outside the national park, where the human pressure basically is high, or it's better, on the other hand, to collect samples from inside the national park for some repatriation program, where, on the contrary, the population size is larger, the animals are less affected by human activities, basically.
Okay, so I may add something to the possible training session that Professor Cioffi already explained. So I think that to produce whole genome sequencing data is important, but it's also important to understand how to analyze this data, starting from raw data analysis to population and demographic analysis. So I would be happy to give training on this if you would be interested. Thank you for your attention.
An applause, please, for Yeah, I understand. This is a sleepy day, and we have two speakers who are actually deep into techniques and species that probably not very many of us understand. Is anybody interested to answer a question? Probably Mas Bayu, so he's done whole genome sequencing. Yeah, please. One name. Okay. Yeah.
Thank you.
I have a question. First, where is the origin population of Komodo from Rinca Island or Komodo Island or Sunda or another island? And second question, after you get the whole genome sequence data of the Komodo island, how to find the functional genes, especially candidate genes for reproduction rights, for growth rights, because yesterday and this is every looking for the functional genes for selection in Paranus species, and you can find the specific genes for reproduction in Komodo. So we use the close family species like Salvador, Paranus Salvador, and fortunately the sequence of the reproduction genes, for example, follicle stimulant and hormone gene is similar. So we will use this sequence information to adapt or to assess in computer because they have similar sequence of follicle stimulating hormone gene like this. So this tell us how to mapping gene mapping, how to find the functional genes in photosynthesis after you obtain the sequence data.
Yeah. Thank you for your question. So answering the first question about the, let's say, sorry, the demographic analysis of the archipelago itself. I mean, if Komodo population derives from Rinca or West Flores population derives from, yeah? Yeah, I mean, you wanted to know if, I mean, one island was colonized before or after. The origin, yes, this is what I'm answering. So it's basically an analysis that you can do using a kind of analysis that it's approximate Bayesian computation analysis, which is a very complex demographic analysis that we are actually performing right now because it was also published recently some statistical pipelines that allows you to investigate these demographic analysis with low genome coverage. So we are doing right now. We don't have the results yet, but yes, this is what we are performing. We are including in the analysis the four main population, which is Flores, Rinca, and Komodo. And for the moment, not to bias the results, we are excluding Nusa Coda and Guillemotang because we have, let's say, few information and the population are very small there. So as a first instance, we are having a look at the main populations. So in order to understand how was the connection between these also, not just now, but also in the past. So yeah, that is what actually what we are doing with this new set of data. And about the second question, that is something very difficult to understand the functionality of genes because the function that you find, sorry, where is it? Yeah. So we were lucky, for instance, here about vomeronasal receptor genes that they were discovered for many other squamate species, and we could compare them to other squamate species and do this nice phylogenetic analysis. So in this case, after the annotation of the genome, we managed to pick these genes because we knew that they might have been different in the Komodo dragon, considering the predation ecology of the species, and actually they were different. So we went straight to the point there. Instead, this about the positive selection of coagulation cascade genes, this was a different approach, because here we did actually a positive selection analysis to see whether any group of genes had internal characteristics that suggest a positive selection of that group, and it was actually for these coagulation cascade genes. But in your case, when you are looking at specific genes that are involved in some physiology related functions, you are strictly dependent on other species, because you need to have a reference, as you said, in other species. Or, for instance, you can have some similar genes in birds, for instance, that you can check maybe if this gene is conserved and look for it in your species. So I would not have a general answer for your second question. I think it's more a matter of this, let's say, analyze case by case to understand what I'm actually looking at. For sure, when you are doing this kind of analysis, you need a very high level reference genome. Otherwise, you take the risk of have bias in your analysis that might lead you to a result which is not actually the biological result you're looking at.
Bisa diterima?
That's the problem.
Yeah.
The full sequence of genome, but we don't know the gene region of the full genome.
Sorry, I didn't get which species of Varanus is this one.
And this Varanus factor for as the different sequence to select computer driven present on the location device.
I see.
Because the sequence is similar.
Yeah.
So what is your opinion about?
I mean, it's similar, but it's not that similar. I mean, also in this, I mean, they are the same genus, but still, when you consider two species of the same genus, it's not really straightforward that you will find the same things in that genome in the other one. So what I would do is not just to consider a single species to check for these genes that you are interested, but I would do a more broad analysis, as we did, for example, here. So here you see we considered many squamates to investigate the vomeronasa receptor gene. So maybe you will find your information comparing your genome to. I would do with all the if there is computational capability, I would do for all the squamates that has a reference genome published.
How to get that genes?
How to catch these genes?
Yes.
The vomeronasa receptor genes?
Yes,
Yeah, in this case, yes. In this case, we were lucky because the vomeronasa receptor genes are very well described in these other genomes. So we did this comparison of just these genes that we fished out from the genome and did this analysis of, which is basically a phylogenetic analysis. So in this case, we already knew the location of these genes in the other genomes. For sure, I would try to annotate your genome also in different ways, maybe also with RNA-seq data, or using different tools for in silico annotation, and then compare all these annotation to check for the coding genes. This part of the genomic world, I mean, the connection between a phenotype and a genotype, I think it's still very far from being standardized in non-model species. So you have to play around with data and eventually find what you're looking for. Okay.
I think the two of you can have a cup of coffee or 2 cups of coffee and chat about
Sure.
really good match. So for your information, Mas Bayu is doing genome sequencing on Indonesian bulls, right?
I'm working mostly in slim array. I'm working in slim array analysis with Illumina machine to detect affordable slim in cattle.
Cattle.
For bulls.
I see.
So it's definitely going to be a jug of coffee for the two of you.
Yeah.
So we have another question in the Zoom room. Mas Suhendra, would you like to open up your camera and microphone probably?
Sorry, can you unshare your file?
Yeah.
There's
There's a question in the chat room by Suhendra. Would you like to open your mic and camera? If not, then Pak Claudio has the answer. You have Rates.
Yes, okay. The microsatellite drivers were found by constructing a genomic library. At that time, we didn't have yet the genome of the common dragons. So the microsatellite drivers were found by constructing standard genomic library in which the DNA is fragmented. The fragments are inserted into vectors, like plasmid vectors. These plasmid vectors were inserted into bacteria, so into electrocompetent cells, and then for cloning. And then the clone plasmids with the DNA insert were extracted from the bacteria, and using known primers of the plasmid, we sequence the insert of the DNA. Then, with these sequences of the inserts, we used probes. The probes were basically the same sequence of the microsatellite that we were looking for. For instance, denucleotide repeats, or a GA, or an AT denucleotide repeats. These probes were marked, and so we mixed the sequence, the plasmid with this probe to find the plasmid that had the insert, the DNA insert with the microsatellite. And so we basically divided, we split all the plasmid with the insert with microsatellites from the plasmid with the insert without microsatellite. And then we sequence, as I said before, using primers of known sequences, all the plasmids that had the insert with the microsatellite. In this case, we were able to characterize the flanking regions of the microsatellites. Some of them could not be used because one of the two flanking regions was too short to design primers. And in the end, we selected these 18 loci. Out of these 18 loci, to answer to the second question, out of these 18 loci, we are actually using 16 of them, because 2 of them, then at the end, the population level results have not be very informative. And these 16 loci, as I showed in the slides, they are reliable enough to determine the population origin, yes, apart from the 2 population of Rincha West Flores, which is very similar. And with microsatellites, we obtain sometimes not very definite results. And that's the reason why for those more detailed, for a higher level of definition, we have to use the whole genome sequencing approach. Actually, after choosing the SNP panel, then one can use a normal real-time machine knowing the sequence of the SNPs to do an analysis using a subset of the 600,000 SNPs that we found, so the 96 SNPs that Alessio will show you before. So,
Swendra, did you hear the answer? I don't know if he's still around, but yeah, I guess we have 10 more minutes to go. If there is any other question, but maybe please.
Welcome to Indonesia, welcome to Sino. So thank you for your interesting presentation. I have two questions, several things to you. When we continue to the question, like you deal with close microcycle for common it's very interesting. But since the common is a very unique animal, especially in India, and to build the good microcycle properly with the good genome, right? So my question is, for the slide that the maybe the people that are participating on other database, JD repository, something like that, how is the update of the component breaker? Is it enough? I mean, like, there's like a lot of sample, the condition of the reference genome. Because if we want to make a good microcycle model, but we need to go back to the reference genome. So my question is it how is it up with the decision? In Europe, really have a possible the so is it need to be improved currently?
not sure I understand the question about
how much the update of the request genome, the whole genome, even the database?
the update of the reference genome
To your it's very hard to get combined, or maybe we need to make it more now suddenly like doing more sample of the data or another collaborator can maybe in the anywhere to compare.
the samples that we have, they will come from developers from the field, from commercial flores. So the DNA samples that we use to characterize the microsatellites came from a mixture of samples from flores, India, and Tahun. We are not spending more time to further characterize other microsatellites because the studies now you can get much more detailed answers if you use SNP studies with respect to microsatellites. So these microsatellites, we didn't go far because the microsatellite test, because they came from natural populations, found them enough to assign single individuals, for instance, of unknown origins that can be bred in a zoo, say Laguna or Zoology, or whatever, to the populations, but to model the school reach West Flores group. Okay? But because these microsatellites have basically they are a limited number of loci. You can go more than say 15 or 20 loci. Otherwise, you have to spend more time, much more time to develop our microsatellites. And you run the risk not to find anyway the level of resolution that you are looking for instance, resolute between giant West Flores, they are very close populations. Then it's more worth to spend time and funds on developing SNP funds rather than continue working on satellites. So once the SNP panel is in place, then it's much easier because with the microsatellites, you have to amplify the microsatellites, and then you have to do the capillary electrophoresis. And then you have to look for the real assignment and construct the genotypes. That for some
biological situation is well. Okay, but in this case, it is the case because we have this problem, let's say, of these two population rich in West Flores that can hardly be detected using microsatellites. While once a SNP panel of say 90, I'm saying 96 because 96 is the number of a place, no? Of a microsatellite. With 96, with this 96, one can do in 20 minutes a PCR on a real-time PCR machine, okay, and have these SNP genotypes for one individual, or for more than an individual if one put more than one primers with different fluorochrome colors in the same way. So it's cheaper basically and it's faster than doing the microsatellites. Of course, the SNP panels, it takes a lot of time to find them. So they are ready for model species, cattle and humans and chickens and so on. But for these wild species, it takes lots of effort to find them. Once they are found, then it's much cheaper and faster to assess the original individuals. And then you can also forget about microsatellites. I'm saying that because, of course,
maybe like here in the Venezuela, that you know, like model in island near the waters, and then we have also collaborated in the zoo,
yeah.
and maybe around the world also have some model. They want to know like
America.
yeah, where does it come from? So the ancestor, something like that. Like,
Population smart.
our stakeholder asking us, like,
Yeah.
can we know, can we find a fastest way to detect, like from where I'm using mine something, like is coming from the island of the dragon, or is it from the Flores?
Yeah.
Maybe if this information or the rapid PCR
Yeah, with microsatellite again, this is something that we are considering for at zoo animals, especially the one European one, because zoos in Europe and the US, for instance, they are not sure where the animals come from. They can rely on what they have been told, you know, so the microsatellites again are used to assess to assess the origin of one animal, for instance, to Komodo or North Flores.
But when the microsatellite result says that the animal is from Rincha or from West Flores, you cannot be sure whether it's from one or the other because they're very close, you know. So microsatellites in our case, they're good to say these animals come from Komodo, from the group of Rincha West Flores, or from North Flores. But then if you want to check, well, this animal is not from Komodo, it's not from West Flores, it's from the group, and you want to say, well, okay, but is it from Rincha or is it from West Flores? Then you have to change marker. And that's the reason why we are trying to develop this NIP panel for Komodo dragons that can also be used by the zoos for a cheaper and faster assessment of the origin of unknown individuals.
Thank you so much. Oh, one little question interesting to Asia. So when you mentioned about the co-article and everything, when the model is unified, and then you need some sort of did you take the example for say conducting the genome sequencing?
No, that is just based on. No, the genome is just that on the paper, on the paper that we design from the dragon, and they know that there is an coagula, the genome, which already is unknown in person. So, and when we found it, like, what is the we say, why we different in the computer, then we connect with this information, these other stories of we then we say, okay, that might be that piece, that something that non-driven as a goal, as a mechanism with own human, but we see it on other names, but it's of course an important question. So growth would be impossible because you would have to count how the gene of the one driver is to
So basically, the source of your sample fluid, the genome analysis is coming from blood or no, here is fine.
no, it's fine. That's the no, it was, but it's fine.
May I know the like the samples that you the results coming from several samples from the different location, is it have a different age of the member or just random samples? I mean, like would the dragon the older one or the young one or just random sampling? Thank you so much.
Thank you very much, Bu Evi. Kenapa? Masih ada saya.
Saya masih mute. Silakan Bu Prof Tia.
Yeah, thank you, Bu Evi. Welcome to Mr. Alessio and Alessio. Nice to meet you both.
Thank you.
Welcome to Bogor, Indonesia. Yeah. I have two questions. One question for Alessio and the second question for Bu Evi.
Oh, terima kasih Bu Prof.
Actually, I don't have experience in analysis, genetic analysis, but I would like to ask about how to find out the Komodo population in nature more accurately. Yeah. What factor are used to estimate the population, and what are the advantage of knowing the Komodo population structure with a genetic analysis approach compared to direct observation in nature, which is more accurate? The second question for Bu Epi. Bu Epi tadi menampilkan gambar-gambar biawak yang bagus-bagus ya, dari serbarannya di beberapa pulau, dari Indonesia bagian barat sampai Indonesia bagian timur. Nah, akhir-akhir ini kan memang banyak yang dibuat dari animal ya, dari hewan, berupa dompet, tas, dan lain sebagainya. Termasuk saya juga punya itu dompet sama tas dari buaya, ini punten nih, cerita saja. Tapi saya ingin tahu, mana yang lebih bagus dari produk from the paranus, itu biawak ya, or dari buaya. Nah, itu. Dan mana yang banyak dipakai, digunakan di industri khususnya untuk wanita dia, tas dan sebagainya itu, mana yang banyak digunakan. Karena ini ada hubungannya dengan nanti mana yang perlu dilestarikan dengan banyaknya biawak-biawak itu digunakan atau buaya itu digunakan sebagai bahan aksesoris untuk para wanita sosialis yang biasa bersosialisasi ya. Mungkin sosialita ya, mungkin itu ya. Terserah ini yang mengkoyotnya Mr. Alessio atau Zoology ya. Because actually I want more know about the genetic analysis ya, and I still learn about that. My subject in the research conservation biodiversity, but not in the laboratorium. Thank you.
Okay, so may I?
You could try, but I guess the questions are actually for Claudia.
Yeah, I will pass it to Claudio as well. In the specific case of Komodo dragon, anyway, just broadly speaking, what I did not underline in the presentation is that genetic tools are important for conservation biology, but they are not better or worse than other methods. So I'm answering generally to your question about which are the advantages or disadvantages of genetic approach, and I would say that these are complementary to traditional approach. So for instance, photo trapping, which is a traditional monitoring approach, gives you a lot of information about, for instance, habitat occupancy or behavior of the species, which is something that very important for conservation biology, but that you do not get for genetic. Or on the other hand, genetic can give you important information, as I show you, on the genomic diversity status, which is not always detectable using traditional approach. So I would say that you do not have advantages in comparison to traditional method, but you have another piece of the story that you can put together to better tell the conservation status of the species. And this is something that researchers are trying to get more also in IUCN, for instance, which does not consider genetic as a proper factor for threat analysis, for threat assignment. So this is my general question about this topic. I will leave the word to Professor Chofi for more specific of the Komodo dragon.
So before answering, I think I have to make sure that the questions are right. Ya Bu, dengan bahasa Indonesia barangkali pertanyaannya bagaimana menentukan lokasi biawak komodo di lapangan kan ya Bu ya? Itu maksudnya pertanyaannya?
Pengambilan di lapangan. Iya, bagaimana?
Oke.
Banding dengan analisa genetik dan mana yang lebih akurat?
Oke, ya. Lalu yang kedua, bagaimana mengestimasi populasinya? Apakah lebih akurat genetik atau secara manual gitu ya Bu ya?
Iya,
Oke.
di lapang langsung, alami. Iya, secara alami di lapang.
So yeah, the question, there are two questions. One is how to find the dragons in the wild. Yeah. So and then the second question is how to estimate population size. Is genetic better than manual study? Yeah.
Okay. The Komodo dragons in the wild,
Yeah.
they are caught with traps. So we use traps, perangkap dengan umpan di dalam, umpan kambing, to trap them, and then when the animal is trapped, I used, and now there is a Yayasan Komodo Survival Program di Labuan Bajo, mereka kerja bersama-sama dengan Taman Nasional Komodo dan BPK SDA. So once the animal is inside the trap, they put a microchip.
Chip, yeah.
Chip, yeah.
Under the chip.
A microchip. And then the population size is estimated by capture, mark, recapture, CMR method.
Yeah. Your research used that method?
Yeah, they is a paper dari Denny Purvandana dan Ahmad Arifiandi di 2004 mungkin. Yeah. Or maybe 2014, mungkin 2014, that they report 10 years study of capture, mark, recapture. So they set up traps. Each time they mark the animal with a microchip, and then they set up traps again. Then they calculate the proportion of animals that in the second trapping session are marked and the ones that are not marked. And then they use the Peterson index, for instance, they use in ecology, okay, to assess the total population size. So in 10 years of capture, mark, recapture, so this is ecology methods, it's not genetic method. Okay? They assess the population size of Komodo, Rincha, Kilimotang, Musakode. Komodo, more or less, is about 1,500, so Ceribus limaratus ekor, di Rincha Ceribus tiganatus ekor, dan Kilimotang dan Musakode mungkin tidak di 100 ekors. So for assessing population size, we don't use genetic method. We can use genetic method to assess effective population size. So basically the number of animals that contribute with their genes to the next generation. They do not include young or old animals. But population size estimates are done using ecological method. Yeah, is that?
Yeah. On your research, use the method you use the method, yeah. Do you know about the structure of the population? How many female, male, and young?
The age structures, yes. This is a work that the KSP has done, the Komodo Survival Program has done. The sex structure is a bit more difficult because there are no yet, like for birds, PCR primers that can unambiguously detect males and females. For Komodo dragons, the primers that are available for doing PCR and then detect the sex still do not give very accurate results. So with the genome, say basically analysis, we are trying to find markers that can provide more precise results. So far,
the
data are for age structure. So how many youngs, how many subadult, adult there are in each islands. Okay? But I can't tell you how many because these are not my data. These are data from the KSP, from the Komodo Survival Program, Yayasan Komodo Survival Program, and the Taman Nasional Komodo. So I don't have them, but you can reach Komodo Survival Program to have information.
One question. How do you think about the why in the Padar Island now we cannot find the Komodo?
Yeah, you can. They have been
Your
recently.
opinion, yeah.
Yeah, there have been recently natural recolonization of animals
Yeah.
in Padar. There are about 12 animals now, something like that.
Because a few years ago,
Recent.
yeah, in Padar, we can find the Komodo, yeah.
Yeah.
But now we cannot find Komodo in Padar.
Yeah.
Padar, Pulau Padar.
Yeah.
Dan
Masih?
sudah rekolonisasi, ada 12 ekor.
Yeah.
Sudah ketemu.
Sekarang
Saya nggak dengar lagi.
yang orang
I don't hear about that.
Orang dari Yayasan KSP mereka pasang kamera trap and to record the presence of Komodo dragon. So there are still.
How about the prey, like a deer?
Yeah.
Still.
Yeah. Also. Yeah. There is also. It's a very recent recolonization. 10 years ago there were no dragons there, but now they are a small number of animals, but there still are. We don't know. They also found evidence of sarang.
Yeah.
Sarang. Yeah. In the forest. Okay. But they haven't found yet hatchlings. So we don't know yet if they are reproducing or not.
Yeah.
But there are some individuals in Padar.
Nice island, yeah, Padar.
Yeah, nice island. Before was even nicer because it was off limits for tourists.
Yeah.
Now lots of tourists.
Many tourists come there. Yeah.
Ya. Jadi sudah ada sampel genetiknya juga dari Padar. Kita sudah deteksi.
Kapan Bu terakhir? Kapan terakhir ada ditemukan?
2000 berapa ya? 18, 19 sebelum COVID itu sudah ada sampel yang masuk ke LIPI ya waktu itu Bu ya. Sudah diidentifikasi lebih dekat ke Rinca daripada ke Komodo.
Padar.
Ya. Nah, berarti Bu ya harus jalan-jalan ke Padar.
Mau.
Okay, probably answer the last question, the first and last question for me. Lebih bagus mana, biawak atau buaya? Itu selera Ibu. Kalau buaya itu dilindungi, insya Allah yang buaya darat tidak dilindungi ya. Tapi kalau biawak itu ada yang dilindungi, ada yang tidak dilindungi. Tetapi yang dilindungi pasti appendix 1, yang tidak dilindungi appendix 2 boleh diperdagangkan dengan kuota. Jadi biawak air ini menurut IUCN juga least concern, malah saya pikir ini bisa saya sampaikan adalah hama sebenarnya. Banyak yang ngeluh tentang biawak di rumahan bahkan. Jadi ketika kita ambil kulitnya untuk bahan baku fashion ya, apalagi nanti yang pakai Syahrini misalnya, itu bisa viral sekali itu ya, harganya bisa sangat mahal. Jadi kalau Bu Tia mungkin setelah Syahrini boleh pakai biawak gitu Bu, nanti bisa endorse. Selera itu. Baik, mungkin ini terakhir ya, Ibu Bapak sekalian. Aduh, Mas Cek silakan, cepat ini. Ya.
Short question is okay, taking something. Just a simple question. My question is related to the Zoology. In the presentation before, you mentioned that you collect 24 samples from population, right? 24 samples of 6 populations. I don't know. like a city for its population and its population.
Yeah, I get it. So as you can see, this is the, thank you. Oh. Yeah, this was the genomic diversity. It's okay, it was a lot of information in one row. So this was the genomic diversity of the populations. And as you can see from here, the range is almost the same for all the populations here that we analyzed, meaning West Flores, Komodo, Guillemota, Nusa Coda, North Flores, and Rincha. And it is nice to see, it's interesting to see that the genomic diversity is lower than some other reptile species, which is critically endangered or vulnerable or extinct, as the pink island tortoise. This is not really, this is just a general thought about it, and information should be taken with care, especially when comparing the genomic diversity with species that live in totally different environment. But anyway, this plot tell us that genomic diversity of Komodo dragon should be monitored anyway in a broader way. And this is the reason why we are doing the same analysis again, not only with 24 individuals, but with 145 to see if we can have a higher resolution pattern of genomic diversity within island.
Thank you, everybody. So 8 minutes to go for 4. I guess I have to wrap up a little bit, but we have learned today from Claudio and Alonso that we move from single locus mitochondrial DNA to multi-locus analysis, and now
we move on to whole genome sequencing. These techniques, we can use them to apply for conservation purposes. And it is a great news that the current Ministry of Forestry is endorsing strategi dan rencana aksi konservasi biawak komodo. Hopefully, it will be signed by the end of 2025. So this might include these new techniques to be involved in direction plan. Yeah. Okay, so I guess that's our meeting today. Semoga ini bermanfaat bagi kita semua. Harapannya di tahun 2026 kita akan bertemu lagi bagi ibu bapak yang ingin mengikuti training pelatihan, bisa tadi ditawarkan mikrosatelit, multilokus, atau bahkan menginjak ke tahap yang lebih canggih ya, whole genome sequencing dan dengan berbagai platformnya. Demikian pertemuan kita kali ini. Saya kembalikan ke Mas Bram. Assalamualaikum warahmatullahi wabarakatuh.
Terima kasih Bu Evi. Terima kasih Bapak semuanya yang hadir di dalam Zoom maupun di offline. Kita sudah sampai di penghujung acara. Kita agak molor banyak, tapi nggak apa-apa. Seharusnya kita selesai jam 16.30, tapi kita sudah sampai jam 15.30, tapi kita 15.53. Sebelum kita tutup, kita akan lakukan foto bersama. So Claudio and Alessio, please open your video. We will have pictures together from Zoom.
Ya maaf, tapi materi saya sudah.
Okay, no problem. Okay, we'll count. 3, 2, 1. Okay, first slide. Okay, we have 3 slides now. It's a second slide. 3, 2, 1. Okay, and the last slide. 3, 2, 1. Oke, thank you very much. Terima kasih Bapak Ibu. Selesai kita untuk acara hari ini. Terima kasih. Ibu Kapus ada yang mau disampaikan? Tidak, tidak. Oke, siap. Kita bisa selesaikan. Terima kasih. Claudio, thank you very much. Alicia, thank you very much for your presentation. Bu Evi, thank you very much for the moderator. It's really good sessions today. So thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Kita foto bareng di luar. Ya. Oh, di sini aja. Oke, siap. Ya. Ya. Siap. Kita ke depan ya.